Good day. My name is Michelle Lavander, and I'm the director of the USC Center for Health Journalism. Thanks for joining us today for our webinar, Do Vaccines Need a Boost? Our conversation today couldn't be more timely. We'll focus on plans by the Biden administration to give out booster shots in response to the widespread emergence of breakthrough infections caused by the Delta variant of COVID-19. Everyone is worried about the Delta variant, but is a booster shot the right path? A debate has emerged about immunity and how to measure it that includes an exploration of protective factors other than the antibody levels that seem to be our current preoccupation. And it's far from clear what scientific consensus will emerge from a Food and Drug Administration hearing on September 17th, just days before the Biden administration was poised to launch its booster effort. The scientific and political debate on boosters takes place at a time when vast swaths of the country still remain unvaccinated. Should our focus be on boosters or on those who can't or won't get vaccinated and who are by far at the greatest risk? Meanwhile, the US booster plans have prompted the ire of the World Health Organization, which just yesterday decried rich countries for using up precious vaccines for boosters instead of helping poorer countries get their first doses to their populations. This is one of the more complex and nuanced scientific, regulatory, and policy stories of the day, one that affects everyone in this country. We're fortunate to have three distinguished experts help us walk through this important story and how to tell it. You can tweet about this webinar at the hashtag COVID vaccine. Our three distinguished speakers are Dr. Dhruv Kular, a contributing writer at The New Yorker, where he writes primarily about medicine, health policy, and politics. He's also a practicing physician and assistant professor at the Weill Cornell Medical College. His research focuses on value-based care, health disparities, and medical innovation. His most recent New Yorker piece delves into the debate that will be the focus of our webinar today. Then we'll hear from Dr. Marion Pepper, an associate professor of immunology at the University of Washington. Her lab shows how cells of the adaptive immune system form immunological memory. And we'll hear from Dr. Monica Gandhi, professor of medicine and associate division chief of the Division of HIV, Infectious Disease, and Global Medicine at the UCSF San Francisco General Hospital. She also serves as director of the UCSF Center for AIDS Research and the medical director of the HIV clinic at San Francisco General. This webinar is made possible by the generous support of the National Institute for Healthcare Management, the Commonwealth Fund, the California Endowment, and individual supporters like you. A word about our format today. We'll be hearing from our panelists first, and then we'll turn it over to our audience for questions. Because we have many people joining us on Zoom, we'll ask you to write your questions into the Q&A panel. You can write us there if you're experiencing technical problems as well. Dhruv, we'll start out with you as we get underway. As if we weren't worried enough about the Delta variant, now we're also hearing about a coronavirus variant known as Mu, which has properties that could allow it to evade vaccine-based immunity. Tell us what might lie ahead with Mu, what we know about both Delta and Mu, and why we don't even know as we plow ahead with plans to launch a national booster shot program about everything that there could be to know about these variants.
Yeah, thanks so much for having us all, Michelle. This is a great opportunity to chat about this really important topic. I think it's helpful to step back a little bit. I mean, we do know a little bit about Mu, but not really enough to speak with any confidence about what it might mean going forward. So Mu is a new variant of concern. It's a variant of interest, not a variant of concern yet, which means we don't quite have enough evidence to demonstrate that it acts differently in important ways from prior variants. So it was first identified in Colombia. It spread to a few dozen countries already, but it really accounts for a small sliver of cases here in the United States. As everyone probably knows, Delta now accounts for more than 95, in some cases 99% of cases in the United States. So I think you're right that there are some suggestions that there are some immune evasive properties associated with Mu, just as we've seen with other variants, but we really don't know enough to to kind of raise the alarm about it. And I think with any new variant, we really want to try to understand three critical properties. The first is its lethality. So if you're infected, how likely are you to have severe disease? How likely are you to be hospitalized or die? The second is kind of immune evasion. And you mentioned that Mu has some mutations that may suggest that it has the ability to evade parts of the immune system in ways that prior variants didn't. And then I think, in my opinion, the most important is transmissibility. And so how likely is it to spread from person to person? And we know that, compared to the original strain out of Wuhan, Delta, for instance, is 5 times, maybe even 8 times more infectious. And that's really, I think, what becomes most problematic. Even when you think about immune evasion, the likelihood of a breakthrough infection, in my mind, rests on a few different things. The first is kind of how likely are antibodies to be able to recognize the new variant. So that's one. The second is how much have our antibodies waned over time. So if you were vaccinated a while ago, and I know we'll talk a lot more about this, you might have a third or a tenth as much of antibodies as you did at the beginning, but there are ways that the immune system rapidly ramps up again. And the third is the viral dose. And so how much of that virus is coming at you. And that's why I think transmissibility and the replication capabilities of these variants are so important. So maybe even more important than their immune evasion properties that get a lot more attention for understandable reasons. And so short answer is we don't know enough about Mu. I don't think it's a reason to kind of change the way that we're thinking about coronavirus at this time. I think our attention really needs to be on Delta and really getting as many people vaccinated as quickly as possible.
Thank you, Drew. And our conversation today about vaccine boosters takes place within a very intense political and regulatory climate. President Biden and Dr. Anthony Fauci have been describing a third vaccine shot as a fait accompli and the new normal, but there are still some really important federal approvals ahead before that could happen, and those have hit a snag or two. So can you walk us through the case for a third shot and the political and scientific calculus at play?
Yeah, I think, obviously we're going to talk a lot more about this, and it'd be great to get Monica and Marion's thoughts as well. There's a lot of controversy around who needs boosters, when they need boosters, why they need boosters. And I think there's even some evolving disagreement between the White House, for instance, and other public health agencies like the CDC and the FDA. And I think the short answer is that there's no slam dunk case. There's not a slam dunk case that, most of the population needs a booster at this time. I think you see that, maybe in people who are immunocompromised, maybe people who are very high risk or very elderly or in nursing homes. I think those are cases in which we should be considering boosters. But for the average adult who's otherwise healthy, we haven't seen data that suggests that immunity is waning or that the vaccines aren't sufficiently protective against Delta at this time. And so I think there's a little bit of a kind of a gamble here, so to speak, in the sense that the White House and the administration understandably kind of wants to get ahead of things. And so they want to be seen as doing something, and they want to avoid a position in which maybe, a few months down the line, we see higher rates of breakthrough infections. We see higher rates of hospitalization among vaccinated people. I think it's unclear that we're going to have that, but I think their calculus is that if that does happen, they want to have been ahead of it. Whereas the FDA and the CDC, I think, haven't seen the data, or at least haven't released the data that suggests that we're at a place where boosters are imperative for most people in the United States. And I will say, I think, as we talk about this, it is just so much more important to have some immunity, either through prior infection or through vaccination, than no immunity at all. And so I think when we're talking about boosters, we're talking about kind of the tail of what we need to be talking about. The real thing is, how do we get people a first exposure to the spike protein in the safest way possible, which is vaccination. But even people who have been infected, they are not going to be starting from a clean slate, no matter whether there are boosters or not boosters for most people.
And what you're saying about this scientific, questioning, really runs counter to what has been the dominant messaging, both out of the White House and in some ways out of the media, which have created a sense of panic about this question of declining antibody levels in those who are vaccinated. Yet rather than being alarmed, scientists, including all of you in this panel, say that the development of something like the Delta variant isn't surprising. And this whole conversation about breakthrough infections has also created a lot of panic. So for the journalists in our audience, can you offer some suggestions on what steps they should be taking to interpret, contextualize, and present the data and all this science and scientific statements while preserving a proper dose of skepticism? And as a writer and a clinician yourself, how do you explain the moment that we're in?
Yeah, I think we're in a really tricky moment. And I think it's one that carries a very high cognitive load for most people. So in one sense, in the past, before vaccines, when coronavirus was everywhere and everyone was immune naive, the calculus was pretty simple, right? It was stay home, wear masks, keep yourself and your families as safe as possible, shut down the offices, don't go to weddings, et cetera, et cetera. Now, a lot of people have to make tougher decisions, and that is a taxing thing. You have to figure out what is worth it, what's not worth it, what can wait, what needs to happen right now. And so it is a trickier moment than in the past. I think in terms of thinking about and contextualizing a lot of what we're seeing, the first thing to kind of think about is, every time we report on a breakthrough infection, every story that we have there are thousands of non-breakthrough infections that we're not reporting on. And so in some ways, that's the challenge, is that how do you communicate that there may be some risk, there may be a rare risk, while not drawing more attention to it than is justified. I found going to the primary data is the most helpful and thing that one can do as a journalist. And I think it's the most important thing. And so even when you think about something like last month, when the White House announced, boosters were probably going to be necessary, the CDC released 3 studies at that time that were in theory the reason, or at least supported that decision. And if you just read the headlines, it's the White House, based on 3 studies in part, is releasing a recommendation for boosters. And if you go to each of those studies, none of them presents a slam dunk case, really. There's one study about maybe there's a decrease in infections or increase in infection among vaccinated nursing home workers. We don't know anything about whether they got sick or had to go to the hospital or how long ago they were vaccinated. There's another study from New York that maybe efficacy against infection dropped from 90 to 80% or something like this, but there was no change in hospitalization, no change in severe illness. And so none of those studies, if you really go to the primary data and look at it, presented a case that, we need boosters for everyone right now. And so I think if there's one piece of advice, I think it would be really going and picking apart the studies that in theory, a lot of these decisions are being based on.
Well, thank you so much, Drew. I'm going to turn now to Dr. Pepper and ask you, you recently told Science that you wouldn't be getting a 3rd shot, a booster. Why not? And why does your expertise lead you in that direction?
Yeah, thanks for the question. And I'm really happy to be here. So I think in keeping with what Drew just said, I don't think that the data so far has shown an imperative need for someone my age who's in good health to get a booster. And I think additionally, we've done a lot of work actually studying immune memory to both natural infections, so people who've had COVID-19, as well as people who've been vaccinated. And what we're seeing is that there is good memory that's forming. There's good immune memory cells that we can look at, both their quality and their quantity. And overall, most importantly, as Drew pointed out, there aren't studies that are showing increases in disease. And I think that that's one of the things that we really have to pay attention to, is what we're talking about with these vaccines is the ability to prevent disease, and they are largely still doing that. So those are the things that have influenced my thoughts on this.
Yeah. What else can you tell us about immunity and the complex way that that other factors are at play in addition to antibody levels?
Sure. I think I'm going to just do this with some slides, which might make it easier for everyone to understand what I'm talking about. So I'm going to put this up. Hopefully you can see this and share my screen. Okay. Great. Okay, so as a professor in the immunology department at the University of Washington, I obviously like to explain things as much as possible so that people can understand the immune system, which I think will help. Let's get this. Can you see that?
Yes.
Great. Okay, so I think it's really important to understand what exactly we're talking about when we're talking about immunity and immune memory, and why it actually works and why vaccines work. And these are some basic concepts that many people already know, but I think they get lost in the conversation about antibodies all the time and the need for boosters. So when your body sees a virus, you mount an immune response against red being the virus. The virus is trying to divide and infect as many cells as possible, while your immune system takes time to ramp up here in blue. And during this period of time, your immune system develops not only more of these cells that can fight off the virus, but they also gain different attributes and characteristics. Hopefully those attributes and characteristics control the virus, the viral load goes down again, and the immune system goes through this process that we call the contraction phase, which literally is contracting that big immune response that you just put all that energy into. And what's amazing about that is you're left with about 10% more cells than you started with, that are cells generally of the adaptive immune system called B cells and T cells, and I'll talk about those in a second. But those cells have inherited qualities during this phase of the response that they can then carry with them for the rest of your life. And so even though they drop down in number, they have these functional characteristics that enable them to respond really fast. So if you see that same virus again or something that's closely related, instead of taking 5 to 7 days to mount an immune response, this immune response happens sometimes in as short as hours. And so that virus never really has the ability to take off and cause disease in the same way it did when these cells were just getting ramped up, learning how to fight this infection. And I think what is lost about a lot of the reporting that has happened is people have really focused on antibodies and just this ability of antibodies to protect against infection, when they're really these multiple layers of immune memory that all act together to control the infection. And just to give you a primer on this, just to make sure that people understand what I'm talking about, there are several kinds of cells that are actually involved in this. There are these B cells, which are the antibody secreting cells. Some of these B cells, called the long-lived plasma cells, can just produce antibodies, hopefully for the life of a person after they've encountered an infection. But you also have these memory B cells, and memory B cells are cells that basically patrol the body and look to see if they can find the same or a similar virus that they saw before. And if that happens, they will rapidly get reactivated to make antibodies. So even though it takes a little bit of time, they are rapidly responsive and can control that infection. Even though you've been infected, they'll control that infection quickly. Now, the sort of often overlooked component of the immune system in this COVID-19 pandemic, I feel, is my favorite group of cells, the T cells. Memory CD4 T cells are cells that can both help these B cells function. There are other types of memory CD4 cells that release these molecules that basically activate inflammation. They cause the production of different antiviral mechanisms. And then the CD8 T cells, which can just basically kill infected cells. So I know it's a lot here, but there are all of these different types of cells that are all doing different things. And I think what I really want to drive home today is that the vast majority of studies that have been talked about have looked at whether these antibodies can persist. But beneath that, there are all of these other layers of cells that can prevent disease. And the reason why they're able to prevent disease is because they're long-lived, they circulate around the body, they basically stay unactivated until they come back into contact with the same virus or something similar. And you can track these over time. This is a beautiful study that was done. And you just need to look at the lines here. This was a study that came out of Emory University, where they looked at cells that were specific for a yellow fever vaccine in people. And they could track, these people were very kind and kept showing up to get tested to see if you could track their T cells over time. And this graph here, these lines, each line is one individual over time, tracking their levels of these cells that could respond to that vaccine. And what you can see is 300 or 400 days later, these lines have largely flatlined. Yes, they've declined from the peak, and there was probably even a higher peak earlier on in here, but they're relatively stable over very long periods of time. And I think even though they're rare, they're still there in the body. The other thing that they've done is they've altered the way that these cells function so that they can get activated really rapidly. They don't need to recreate all of these different programs that let them both have different functions and divide and do all these things. They already have that. That's why it's called memory. They've retained that ability. And that lets them respond really rapidly. And then last thing that they do that's also really important that makes it very tricky to study these cells, and I think is part of the problem, is a lot of these cells go into different regions of the body. They're not just circulating through the blood. Some of these cells will go into the lungs. Some of them will go into the tissues. And by that, I mean the digestive tract. This was a beautiful paper from Donna Farber's group where they actually looked in autopsy patients. They looked in autopsies for T cells in tissues, which is, of course, the only way you can do this in people. You can't just take random tissues and look for these immune memory cells. So the point of this is that we are measuring antibodies, and antibodies may be decreasing, but there are all of these other parameters that are hard to find, and they're rare and they're difficult to track, that we have a really hard time looking at so that we can say, yes, once we get to this one point in waning of immune memory, that's when we need to be concerned. And I think that's part of the problem. And so one of the things that my lab has been doing is trying to really study this over time in people who had both infection and have been vaccinated. And I'm not going to go into all of the detail here. I think it's just important to look at the colors here. Each one of these lines going across, this is just what we call a heat map. And so each one of these lines represents a different immune parameter. All the people on the right here are people who had a natural infection with COVID-19 early in the spring. All these people over here are healthy controls. Red means high, blue and white. White means zero, basically. Blue means low. These are people who are healthy controls. So if we look for all of these different immune parameters and all of those different things that I just told you about that form this functioning system, it's an immune system, it's not just an antibody. You can see that all of these people have really high immune responses and across multiple types of immune responses and immune parameters. And I think that's really important to tell. And so what we've been doing now is really looking at how do antibody levels compare to all of these different factors that I just told you about, and how do we relate these two? And what you can see over here is if we look over time, this is 1 month, 3 months, and this is 9 months, we can see that antibody levels are falling in people who had a natural infection with COVID-19. There's no doubt that they're waning over time. And more people sort of fall under this cutoff as time goes on. But if we start and also the other thing we can do is look to see whether those antibodies are capable of blocking infection. So we just take the antibodies out, we put them in a dish with the virus, can they block infection? And if we look over time, yes, that is lost to some degree over time. By the time you get to 1 year after infection, about 50% of those people still have antibodies that can block infection. But if we start to actually look and see what was a little bit more concerning to us, how this impacts the response to variants, we can also see this is a variant that originated in South Africa. It's called the B1351. It doesn't really matter, but we can see that those antibody levels are even lower and they're able to neutralize even less against the variants than they were against the original. So this should really make us concerned. And I think this is what a lot of the news was reporting, is that if there aren't these antibodies and they can't actually block infection, then maybe there's going to be this loss of immune protection. And I think what really needs to be remembered in here is that antibodies are like the force field, and if you've seen The Incredibles, that is sort of sent out to protect the family. But once the infection gets beyond these antibodies, if they wane, if there are holes in this force field, there's this whole team of superheroes that's left underneath that force field. And so what we decided to look at was, what do these individual parameters look like? And can we find B cells and T cells, long-lived memory B cells and T cells that I just told you about, that can actually respond to the virus and respond quickly? Because if we can find those, we're going to be less concerned about those antibodies waning. And what we decided to do was look at B cells and T cells that could respond to both the original virus and to the variants. And I'm not going to go through all the data today. I'm just going to give you a brief snippet and hopefully give you some optimism. And hopefully this will explain why I don't feel like I need a boost right now. But we can track these B cells that have been generated in response to either infection or vaccine over really long periods of time, and then test to see if they can make functional antibodies. And we can do this either in people who haven't been infected or people who have been infected across the bottom here. And all I want to show you here, and I know there's lots of dots and none of these words really matter. I just want you to look at the shapes on these two plots. So in this top plot here, you have what we call the healthy control. So this is someone who has never seen COVID-19. These cells in the upper left-hand corner can respond to the first type of SARS-CoV-2 that was encountered in the US, the Washington strain, we call it. These cells down here can respond to a variant. There are very few of each in this healthy control. Most of the cells that are in there can actually respond to both. But after you've had an infection, and keep in mind, this is before vaccination, you can find this bigger population. These sort of contour plots represent a larger population of cells that can respond to both the original strain and the variant strain. So even though those antibodies are declining, even though you don't have that same capacity to prevent infection, if you do get an infection, these cells, and whether it's the original infection again, or one of these variants, these cells here can rapidly respond and lead to protection. After vaccination of our healthy control, you can see that now these people after vaccine, after having 2 doses of vaccine, have these same big populations of cells that can recognize both the original SARS-CoV-2 virus that came into the US and this variant 351. So what this is telling you is that, yes, we're seeing declines in antibody, but there's this underlying layer of memory B cells that are sitting there that are ready to respond. There's lots of them, and it doesn't matter whether it's the original strain or the one of the new variants. We can find these cells against those, and they're going to get reactivated, and those will control against disease. And it's really important to remember that that was sort of the original goal of the vaccine, was to prevent people from getting sick and from dying. So, yes, immune memory is waning against some of these parameters, specifically some of the long-lived antibody titers that people are measuring. But underneath that, there are multiple layers of these responsive, diverse, and protective cells. And that is sort of my argument for why I will not be getting a booster right now.
Thank you so much, Dr. Pepper. We're going to turn now, this is so helpful, to Dr. Monica Gandhi, who's going to delve a little further into this whole question of our immune system and how it works and how it will apply for this this conversation about boosters. I think you're muted.
Sorry about that. That was a great summary because you are a professor of immunology at UW, and it's actually anything I've been doing to talk about immunology for the public is taking the great work that you and others have done during this pandemic and try to explain it simply for the public, because that's exactly right. In terms of how the immune system works, it's not just antibodies, and we're really seem to be doing a disservice if we just talk about that. You know, we have 9 vaccines now in the world, and we have 3 in this country, and the 3 in this country involve the spike protein in some way, basically coding us as humans, coding for that spike protein, getting a little bit of genetic material, either through Johnson & Johnson, which I know keeps on coming up, that's DNA genetic material, versus the 2 that give us mRNA genetic material, Moderna and Pfizer. All do the same thing though. They make your body produce the spike protein, and then you raise a really strong immune response against it. And the important thing is you raise a strong and diverse immune response. You raise B cells, and we just really heard about B cell memory, and you raise T cells. And as an HIV doctor, I know how important T cells are because HIV infects the CD4 cell, and they provide long-term immune defenses against severe disease from viruses. And yes, your B cells will go into your memory. And of course, antibodies will wane because we don't want our blood to get all thick with all the antibodies we've ever seen in the world. But then what will happen is your B cells will just, with help of the T cells, will produce those antibodies right back up if we see the virus again. So T cells are important because they protect us against severe disease. And you just heard from Dr. Pepper how long those T cells last. It looks like they have the half-life of what you get after yellow fever vaccination, which is one time only, lifelong. So it may be that you get, because T cells protect us against severe disease, you may get lifelong protection against severe disease, but certainly if your T cells aren't fading, that's why all the studies are showing us consistent protection against severe disease. And what Dr. Cloder said is correct, that the studies have not shown convincingly that the protection against severe disease is massively down. Remember that the trials, and no too much to go over right now, but all the trials, including Moderna, Pfizer, Johnson & Johnson, didn't just measure antibodies. They took the trouble, they used immunologists like Dr. Pepper, and they measured T cell responses. This is column 4. They measured T cell responses that you got to the vaccine. So it's not just antibodies, they measured documented strong T cell responses from the vaccines. And in the trials, almost 100% protection from severe disease. That's your really amazing T cell response from these vaccines. Yellow, down the line, all of them, almost 100% protection from severe disease. So the reason that T cells will work against variants, which Dr. Pepper just told us, is that essentially the spike protein is yay long. I mean, it's really simple. And you have these different variants. Only alpha and delta have taken hold in this country, and now it's all delta. And I agree with Drew that mu is it's of interest, but not of concern, and agree with the WHO and the CDC on that. But delta is so transmissible. That's the most evolutionarily sound thing a virus can do for itself. We don't like it, but that's exactly what it's doing. It's making itself more transmissible. That's evolution. And it's likely the most transmissible variant we're going to see. I don't think anything's going to be able to compete with this. We got to get on top of the delta, and that's through immunity. And remember, the spike protein is this long, and it's that simple. The delta variant has 11 mutations, but you have like hundreds of T cells, or at least 100, maybe 87 in one study, line up across the spike protein. And so you take away 11 of those mutations from the delta variant, make 11 of those T cells don't work well, you still have 75 left. So you get a very robust T cell response against the spike protein, which is why we haven't seen T cell immunity decrease to the variants from the vaccines or from natural immunity. And so is our vaccines waning in effectiveness? Exactly as Marian said, it really actually has to do with if you think that antibodies waning and then you taking a minute to make those antibodies. And so you may get a little mild infection before as you're making up your antibodies again from your memory B cells with help from your T cells. If you consider that waning effectiveness if you get a minor infection versus the protection from severe disease. And remember, vaccine effectiveness, this is from Yuge Sebek, is not just it's not just about one thing. It's not just about the vaccine. It's about how long you gave the doses apart. Giving longer time between doses usually increases your immunogenicity. It is about if you are exposed to a lot of virus, so these mild infections, there was quantification by the New York Times yesterday, David Leonard, 1 in 5,000 in places with high circulating virus, and then minor infections, 1 in 10,000, like the city in which I lived, San Francisco, where the circulating virus is down. It depends on you as a host. So if we're going to give boosters, giving to immunocompromised individuals has been approved, makes sense. We've always done that with immunocompromised patients to build up, they may need a little more to build up their B cell and T cell memory, but it's not just one size fits all because it's so complicated when we measure vaccine effectiveness. And really the important thing is the studies, including those 3 CDC studies that Drew told us about, one from LA and New York, for example, showing really high persistent effectiveness across the world against severe disease. Israel, England, Canada is not on here, US, Qatar, persistent effectiveness against severe disease above 90%. So, you could say something really dramatic like, are we going to give all immunocompetent Americans a booster to prevent a cold? Since the massive effectiveness against severe disease is maintaining, and not give it to the rest of the world. Because one thing I want to remind us is that 2% of the doses that we have in this world have been given to low-income countries. So we are actually probably sitting on about 1 billion, at least according to the Duke International Research Center, about 1 billion surplus doses. We have enough. World Bank has estimated that rich countries have enough to give 5 doses to each of their rich people in their rich countries, while people are truly, truly dying of a vaccine preventable disease right now in sub-Saharan Africa. So I know there's geopolitics here, but boy, I can't imagine this is going to be good for our geopolitical position to give vaccines out, a booster to every American for a cold. And so just remember that just as a visual of this, of course your antibodies are going to go down because you can't keep it all in your bloodstream. It's just too much, it's too thick. But the next time you see it, your memory B cells are going to happily produce more antibodies with help of your T cells. And importantly, those antibodies from Oregon and University of Pennsylvania studies are adapted to the variants that you see. So those antibodies are, because it's like a blueprint for a house. If you need an extra brick over here, the house is going to adjust. And so those memory B cells actually produce adaptive antibodies that look like what they need for the variant, mu or delta. So that's even more complex and real than giving a third shot of the ancestral sequence of the virus. And so right now, boosters, as of today, are only approved for immunocompromised. Again, like Michelle said at the beginning of this, September 17th, this FDA is going to review data for who else would need a booster. Maybe older people, maybe people in long-term care facilities, but certainly the whole population with these studies doesn't seem indicated. And finally, I do want to mention that we got a little confusion from the CDC at the end of the month in July about vaccinated people being equally as likely to spread. That's not true. Actually, your immune response will kick in. Your antibodies will come into your nose. You already have T cell islands in your nose. That was not true. It was based on a single time point from a single study. But there's been really good studies since that. This was a Singapore study that shows that if you're vaccinated, what happens even if you have a viral load with Delta that's equally as high for a minute, your immune response comes in and brings down that viral load. So the green line shows the viral load comes down much more quickly than if you're unvaccinated because you have adaptive immunity from your vaccine. So isolating someone for 10 days is not necessary if you're fully vaccinated. You're going to be infectious for a lot shorter period. And then importantly, the immune system's going to try to kill that virus. So it's going to get sick really fast. And so this was a study from the Netherlands that showed out of 161 Delta breakthroughs that the virus was not as infectious because it's being attacked by the immune system. So when you put it in culture, it looks more sick. It's not as infectious. So I'm very much less likely to spread if I have a mild breakthrough with a vaccine on board than if I'm unvaccinated by culture data and by viral load data. And this is a Singapore study. This is as good as we get. We really need to do careful contact tracing, but at least in the time of Delta, if you go to this website up at the top and go to Singapore, where they do careful contact tracing and put in the words vaccinated and asymptomatic, our postdoc looked at this, we can only see one infection from a vaccinated asymptomatic person. So putting back masks for vaccinated asymptomatic people in all settings seems a little overkill, and I'm very comfortable being around my vaccinated people who I know who are asymptomatic. And then finally, if we're going to decide on boosters, I would take this data, take the CDC's data on severe breakthrough infections. They have, I needed to update the slide. We have about 7,500 severe breakthrough infections. It's the largest data set of breakthrough infections that we have, but they haven't stratified the data. Take this data, tell us how many are over 70, tell us who got Pfizer versus Johnson & Johnson versus Moderna. Maybe they're all Pfizer, because Pfizer actually, if you give it too short of a period, the antibodies wane more quickly. Maybe they're all have multiple medical conditions, and give boosters to those who are at risk for severe medical conditions. That is what I can see as kind of politically the way out of this controversy, where the FDA says one thing, the White House says one thing, the CDC says one thing. What we could do is just all together say the FDA is going to review this breakthrough data and come up with some nice, very sensible way of decide who needs a third shot. And that's what I would suggest. And that I will end there so we can take questions.
Thank you so much to all of you. So just a reminder to our audience, we already have some questions in the Q&A, but if you have questions for these three really amazing speakers, please put them in the Q&A panel. And as we start to call on you, I just want to turn back to Drew, who got us started. Now that we've heard all this from all three of you, I just want to ask you, what problem are we trying to solve with the booster shot, and will it accomplish that?
Yeah, I think that's really the fundamental question, not just for the booster, but kind of the end game of the pandemic is, how will we know that it's over, and what problems are we trying to solve? What are we trying to prevent? I mean, I think from a layperson's perspective, the rationale for masks or business closures or capacity restrictions has changed over time, right? At the beginning, if you recall, it was to prevent health systems from becoming overwhelmed, and then it shifted to kind of making sure that older people or more vulnerable people weren't at high risk, and then those people got vaccinated, and now it shifted to, well, kids still aren't vaccinated. I'm not saying that any of these are not good rationales, but we should be clear, and we should have a public conversation, but also, the federal government, the administration, the CDC, the FDA, we have to be clear about what the goal is, because if you come to a conclusion that the vaccines aren't sufficiently protective, then there's no end, right? Some combination of vaccination and naturally acquired immunity through infection needs to be good enough at some point, and trying to understand what that point is depends on what our goals are. So, I'm a proponent of having, kind of clearly defined metrics. I know I've talked to Monica about this in the past as well. And, one thing that seems to make sense is making COVID into something like the flu. And we've discussed that as well. And whether or not you agree with that level of morbidity and mortality, it kind of does suggest that that's, our society's revealed preference of the level of, risk we're willing to take in order to be able to be with other people in large settings and unmasked and so on. And so, that requires thinking about, what is the path to getting to a place where we have, 100 hospitalizations a day or several thousand cases a day, whatever, the average number of flu cases and hospitalizations are over the past decade. That may not be what we end up deciding, but I think in the absence of having a conversation about what we're trying to accomplish in this next 6 months or a year, it's very hard to understand whether people should get boosters, whether we should have indoor mask mandates, et cetera, et cetera.
Thank you. I'm going to turn to our audience. I'm sorry, Monica, do you want to say something?
I would love to add to that, because I think this is exactly, Drew is exactly on target, that essentially my impression for why the White House initially said they wanted boosters was to actually increase the antibodies in the nose of people who had been vaccinated already, to decrease transmission because of the Provincetown data that showed, just a single point in time, that the vaccinated and unvaccinated could have the same viral load, at least by an inadequate measure. But you really need microbiologic data to see if that viral load is truly infectious and if it comes down fast. And the idea that you can actually block transmission completely, it means to me that there is not a complete acceptance in the United States, as opposed to other countries, that this is going to be an endemic illness. And endemicity of a virus in infectious disease principles means that you actually live with the virus, like we live with rhinovirus and adenovirus. But what we can't live with is severe disease. It's actually why we shut down society. It's why we did mask mandates, all of that. But we can't live with severe disease. But the decision about whether we can live with a defanged virus, a virus that has been stripped of its ability to cause severe disease, and this becomes the 5th circulating coronavirus that causes colds, that is a decision that Europe has already made. Denmark is going to open in 2 weeks. Ireland's going to open in 3 weeks. UK opened on July 19th. EU is in charge of 400 million people, and they have said no third shot right now. They have accepted endemicity. I think we have not accepted it. And then one final point, because someone asked about long COVID, is that there's a very nice study from the Lancet that looks at post-vaccination mild infections. And there doesn't seem to be any appreciable rate of long COVID with a mild infection. And that really makes sense pathophysiologically, because how you get long COVID is really from the virus spreading. And that's what your immune response is exactly preventing from happening, and also from a massive inflammatory response. And both those things are brought down by the immune response to vaccination.
Yeah, one other thing I just add on that, Monica, is, the Provincetown example is why it's so important to understand the context and go to the primary data, is because it is hard to imagine a situation that is more conducive to viral spread than what Provincetown was during the first week of July. I mean, it was huge parties, it was raining, clubs were packed indoors.
No masks and intimacy.
Exactly.
Intimacy,
And under those circumstances, you might say the vaccines did incredibly well, actually. No one died. There were 7 hospitalizations. There were 60,000 people there. And without understanding that context, you tell a very different story about vaccine efficacy and what type of place we're in when it comes
and
to
also the
pandemic.
rate of symptomatic breakthroughs, which estimated yesterday 1 in 10,000, 1 in 5,000, not 70% in that high stress situation, right?
And kind of following on this, we have a question from Nicholas Jervis saying, Dr. Pepper, why are so many studies focused on antibodies if, as you point out, so many other immune factors, including memory, are at play?
I'll give you 2 different answers. The first one is they're easier to measure. It's a lot easier to take a blood sample and measure antibody levels than it is to do these very detailed, in-depth analyses of individual rare cells that you need to pull out of the body and study. So it's easier to measure antibodies. But also, I don't want to underplay the fact that antibodies can act as this protective shield. So the thought is, I think, in a lot of people's mind is, if you have that first line of defense and you can keep the virus from breaking through that, then everything's going to be okay. But the reality is, even if that first line of defense goes down a little bit, you still have all of these backup players and everything's still going to be okay. So I think that's why.
And Will Cushman says, the question is for Dr. Pepper. When you talk about the other immune system mechanisms that can prevent another bout of disease, do you mean preventing infection, i.e. a breakthrough case, or preventing symptomatic disease? This is something I get confused about, especially since SARS-CoV-2 and its variants can cause asymptomatic infections, even in unvaccinated people. Also, are declining antibody levels something we see with other common viruses and vaccines against them?
Yes, so I'll answer the second question first. Declining antibody levels are very typical with a lot of viral infections and other types of infections. So that we'll just get out of the way. Thank you for asking that breakthrough versus symptomatic disease question. I think what is really important to remember is that T cells largely don't even get activated until you've had a productive infection, until that pathogen, that virus or bacteria or whatever it is, has actually successfully invaded your body. So the immune system is actually built, that immune memory is built to be reactivated upon what everyone's calling a breakthrough infection, and then prevent disease. And that is basically how it's evolved, is to recognize infected cells, especially for the CD8 T cells I brought up. They recognize infected cells. They can't recognize just a virus floating around. They can only recognize infected cells, and then they kill those infected cells. So I think it's a really important point, and that is what prevents disease.
And we have a question in the chat. Dr. Gandhi has mentioned before that she has sent immunocompromised patients for testing for other types of immune cells after vaccination. How difficult is it for these patients to access such forms of testing, and does insurance typically cover it?
Yes, just like Dr. Pepper said, the reason antibody studies are so common is because they're really easy to do, and many labs have done it. Actually, doing T cell immunity takes a true immunologist like you, and we have some of those immunologists here. So actually what ended up happening in these patients is because I have connections with my friends who do T cell tests, like Dr. Henrik and others at UCSF. I got T cell immunity measured. It wasn't a commercial test. So I think, it's interesting because Congress, Stephen, there was a congressional initiative to say we should develop some commercial tests for T cell immunity. Only an immunologist can tell me how hard that's going to be to do to really get a true correlative protection, because antibodies are not a true correlate.
And we have a question from Sally James. She says, Dr. Pepper's presentation was wonderful, but if vaccines don't halt transmission and students without vaccines are indoors in schools, the vaccine is not helping dampen down transmission to the unvaccinated. Could you speak to that?
I think actually Dr. Gandhi already answered that by showing the data that shows that not only is the viral load going to be controlled faster, so there is going to be less transmission over days, but some of those viruses that they were able to take out of people who'd been vaccinated were less able to infect cells. So I don't want to say that there isn't going to be less transmission in people who've been vaccinated. There is definitely going to be less transmission. And I think those studies are coming out now that are really showing that very clearly.
I would say really that, to me, that comment that they don't reduce transmission really again belied a knowledge of virology, really. I mean, the virus is being, and immunology together, of course the immune system's attacking it. So the virus, even our PCR tests are very limited. They can pick up dead viral particles or pieces of the virus. But of course the virus is being attacked, so transmission will be reduced. Yeah.
Meg Wingertner says, in many states, overrunning our hospitals is still a real possibility. How should vaccinated people act if the risk is low, but the system can't really absorb any more patients?
I mean, I can say what I'm doing, which as a vaccinated person, I'm still wearing a mask in largely populated spaces. If I travel or if I'm indoors, I still wear a mask if there are a lot of other people around. And I think Dr. Gandhi can probably talk to this more, but I think having the vaccine and then additionally masking to prevent transmission and spread is probably the clearest way to do this.
I mean, this is what you just said is why it's so hard in our country, because what happened is if you're in Denmark right now at a uncomplicated political environment where the vaccination rate is up to 80% and you're going to open in 2 weeks, the reason you feel like that is because the hospitalizations are low. And the hospitalizations are low in many places in our country where there's a high vaccination rate. And in other places where adults who are eligible didn't vaccinate, the hospitalizations are high with the Delta variant. And that is so painful and so variegated to see that response around the country. And it's why the CDC does have a hard task, sort of messaging for the whole nation, but to message that those who are vaccinated need more of a life raft as opposed to those who are unvaccinated are the ones we have to work on and really try to get vaccinated, can erode confidence in the vaccines. The booster conversation can erode confidence in the vaccines. The best thing is to get unvaccinated adults vaccinated. And then yes, inside I would wear a mask if I was in a place with high rates of transmission.
And one of our audience members asked Dr. Gandhi if you're going to get the booster shot.
Only if my hospital makes me. There is going to be criteria that come out. I'm positive the FDA is going to be reasonable about this. Again, people in long-term care facilities, people who are older, possibly with multiple medical conditions. One criterion could be healthcare workers, because even getting a mild infection, I can't afford to stay out of work, and I can't also spread to vulnerable people. So that group has been floated in these conversations the last couple of days as the conversation has shifted back to doing a targeted approach for boosters of healthcare workers. So if my hospital makes me, I will, but I wouldn't otherwise.
Thank you. Abby St. George says, I've heard people say I've had COVID, so I don't need the vaccine because I have antibodies now. Do we know if vaccines are more effective than natural immunity? It sounded like in the presentation, the response against variants was similar to those with the variants and those who just had COVID.
I think there have been studies that have shown that there are some studies that have said that the response to infection, the immune response and immune memory to infection is better than the immune response to vaccination. There are other studies that have said the opposite. I think it's really important to remember that anytime you're comparing a person who's survived COVID and has developed immune memory, there's already survivor bias in that population, and you're not really comparing apples to oranges there. So I think those are difficult things to actually make that direct comparison. And it really doesn't take into account what the body has to go through and the risk that you're exposed to by enduring that natural infection to get there.
Thank you. Thank you. Well, I want to thank our really amazing speakers for their time here today, and for all of our audience members for their great questions. I want to mention that we have a feature, a regular column we have called You Asked, We Answered. And I know there's more questions here than we had time to answer, but we'll try to get to a few of them through that column as well. And just want to mention that we'll be sending you all a quick survey to ask you about this webinar and your ideas for future ones. And we'll ask you to take a moment to fill that out. We'll be archiving this webinar a little later today. And should you want to support our work, ongoing work with the Covering Coronavirus series, please just send a text to 41444 and type the message CHJ for further instructions. We welcome your support, and we want to thank everybody for joining us today.